Assalamu alaikum. This video is an introduction to Chapter 5 of your books, Human Genetics. In the previous lessons, you have learned about the experimental methods used to study the transmission of a trait and to find the real genotype of a given phenotype. But when it comes to humans, the study of the transmission of the human traits cannot be done by experimental methods that are based on trying different types of crosses. Experimental methods cannot be applied in humans for many reasons, such as the smaller number of descendants, the time separating two generations, and the ethical issues, of course. So, if a couple wants to know if there is a possibility to transmit a chronic disease to their children, scientists will probably use statistical methods and will mainly depend on information provided by the analysis of the pedigree of this couple. The pedigree, which is also called the family tree, is a chart that shows the appearance of a specific phenotype in a certain family. The pedigree can help us understand the transmission of a trait in past generations to allow predict its transmission in future generations. How? That's what you will learn about in this chapter. But first, and through this introductory video, you will learn about some key terms related to the pedigree and its construction, distinguish between autosomal and gonosomal genes, and use the pedigree to prove an allele to be dominant or recessive, because you'll need these basic information throughout this chapter. In a pedigree, the family members are represented by symbols. Males are represented by squares, while females are represented by circles. Married couple are connected by a horizontal line that represents mating. But if a couple is joined with two horizontal lines, then this represents consanguineous mating, which means that the couple are close relatives. The children of a couple are connected to the mating line by a vertical line, and all brothers and sisters emerge from the same horizontal line arranged from left to right by their date of birth from the eldest to the youngest. A fetus that is not yet born is represented by a rhombus. Fraternal twins and identical twins are distinguished from other siblings by the following symbols. In a pedigree, generations are represented by Roman letters, while members in every generation are given numbers. And by convention, if a pedigree is studying the transmission of a certain allele, then all colored symbols represent members that have this allele expressed on their phenotype, while those empty symbols represent members that do not express the allele under study on their phenotype. The most common standardized symbols that are used in a pedigree are shown in document E, page 93 of your books. Now, after you know how to read a pedigree, you must be asking, how can a pedigree help in studying the transmission of an allele? And what information does it provide? Actually, a pedigree allows to establish an accurate or a probable genotype of the individuals in a family. It also allows to estimate the probability to have a child with a certain hereditary trait, which we also refer to as the risk. A pedigree also allows to study the mode of transmission of hereditary traits. From the analysis of a pedigree, you can find out if an allele is dominant or recessive, and whether a certain gene is localized on the autosomal or gonosomal chromosome. Let's start by proving alleles to be dominant or recessive using pedigrees. Consider this pedigree of a family with some members being affected with sickle cell anemia. Do you think that the allele for this disease is dominant or recessive? How to know this just from observing the pedigree? Consider the affected male too of the second generation. This affected male must have inherited at least one allele for anemia from one of his parents. But both parents are healthy, and the allele for anemia is not expressed in neither one of them. This indicates that the allele for anemia is recessive and masked by the dominant normal allele. Now, this is another pedigree of a family with some members affected with migraine. If you observe the pedigree carefully, you can see that this affected couple who give birth to a healthy male, 3. This time, this male has at least inherited one normal allele from one parent, but both parents are affected with migraine, which suggests that the normal allele is masked by the dominant allele for migraine. But actually, In case of dominant genetic disorders, it's not that common to find a couple that are both affected with the same disorder to choose to get married. So you will most probably be given the following pedigree. You will certainly ask, how am I supposed to prove the allele to be dominant with no affected couple having a healthy child? Well, there's another strategy to follow. A dominant disease that is usually rare in the community exists in high percentages in a certain family, in such a way that you can find at least one affected member in every generation. So whenever in a pedigree you find that every affected member has one affected parent, then this suggests that the allele for this disease is a dominant one. The pedigree analysis can also be helpful to determine the location of a gene, whether autosomal or sex-linked. Remember that in humans, each cell normally contains 23 pairs of chromosomes. 22 of these pairs are called autosomes, and they look the same in both males and females. The chromosomes of 23rd pair are the sex chromosomes. These chromosomes differ between males and females. Females have 2 copies of the X chromosome, while males have one X and one Y chromosome. So the genes that are carried by the autosomal chromosomes are called the autosomal genes, while those carried by the gonosomes are called the gonosomal or sex-linked genes. Since both males and females have the same autosomal genes, then the transmission of those genes will be the same independent of the gender of the individual. But this is not the case for sex-linked genes. You see, when it comes to sex-linked genes, males and females are not the same. A female has 2 X chromosomes, as we said, that are homologous and carry exactly the same genes. This means that for any gene carried by the X chromosome, a female always has 2 alleles, just as you can see in the following example. A1 and A2 are 2 alleles for the same gene. The genotype of this female concerning this gene is written as follows: XA1, XA2. But males do not possess homologous gonosomes. Actually, the X and Y chromosomes possessed by a male are partially homologous. What does this mean? It means that both chromosomes have a homologous part with same genes, but also have parts that are non-homologous and have different genes. So in males, only if the gene is carried by the homologous part of X and Y, the male will have 2 alleles for this gene. In this example, the genotype for this male concerning this gene is written as follows: XB1, YB2. But if the gene is located on the non-homologous part of the X chromosome, then the male would have only one allele for this gene, and the genotype of this male concerning this gene would be written as follows: XC1Y. You see, the Y chromosome doesn't carry any allele for this gene. It is only carried on the non-homologous part of X. Similarly, if the gene is located on the non-homologous part of Y chromosome, then the male would have only one allele for this gene as well, and the genotype of this male concerning this gene would be written as follows: XYD1. This time, the allele for the gene is only carried by the Y chromosome and not found on the X chromosome. So far, those are the basics that you need to know before starting this chapter. In the next documents, you will learn how to use a pedigree to determine the location of a certain gene, whether autosomal or sex-linked, linked either to the non-homologous part of X or the non-homologous part of Y, and finally calculate the risk of an individual to be affected by a certain genetic disorder. Assalamu alaikum.